So maybe you now see the trend of the presentations here. It's sort of starting from basic research, moving over to small biotech companies rapidly growing to a larger company called GE Healthcare and Life Sciences. Slightly bigger, we're about 5,000 employees. We're also actually rapidly growing, but we represent sort of the more commercial aspects of the protein science in the region. And just to give you a brief background, we have about 20,000 different products covering more or less the entire space, reagents, chemicals, equipment, chromatography resins, plastic bags, everything you need to discover, to do research, to develop and to manufacture proteins for academic use or for industry use. So it's a very broad spectrum. I will not try to cover those 20,000 products and what we have done. So I picked up a few examples just to think about how we connect to the trends we see in the global perspective and also the development ongoing in the region. We have about 5,000 employees in GE Healthcare Life Sciences. I'm heading R&D. We are about 1,500 in the Uppsala region and in Sweden, and about 500 in R&D. So it's a fairly large investment into the next generation of products in this region. I have also groups in US and in India, but the majority of the research we have in the Uppsala region. So I'm going to give you a few examples of the technologies, but want to connect to the morning session a bit, what we discussed. So when we look about the challenges for the future, we see that there's a huge unmet medical need in a global context. And what can we do to serve that need as a technology provider? We want to provide new technologies that reduce the cost for health procedures. And we also want to provide new technologies that improve the quality of those drugs. And also what we can do to increase access to these drugs. Can these drugs or treatments be used also in the developing countries like India and China, and also into remote locations? So this is actually a $6 billion investment up until 2015 to work on more technologies to provide sort of tools for this purpose. We call this health imagination because it's up to our imagination to provide these tools to increase the health in a global context. So some bits and pieces of what we do. I said we have sort of this broad coverage. We have tools for research and discovery for more academic settings. It's mainly focused on protein technologies, but also technologies for small molecule-based drugs. It's also analytical technologies, like the BioCore assay is used very extensively in these settings. And we also gone into the same sort of arena that Christian is working on for the cell technology. So we have tools, equipment to produce cells, and we also have cells that can be used as tools for preclinical predictions. We also have a large number of consumables that you do use in your day-to-day laboratory. And then we have the other portion of the company, which is dedicated more to the biopharmaceutical industry. All those guys that produce all these monoclonal antibodies, for example, and all the other protein-based drugs. And also in this arena, we cover everything from very, very small scale, like this tiny, tiny scale, to this huge scale. So scalability is one big concept for our company. And we also have everything covering from cell cultivation, upstream technologies, the purification, the downstream technologies, and the protein analytics, so covering that entire flow of development. And then, of course, we have the service, the break and fix stuff that goes along with all our equipment. So that is a bit of the overview, and then I'll go into some deep dives. So we talked a lot about these monoclonal antibodies. And the good part of the story is that monoclonals are one end of the molecule identifies the target. And those are unique for each monoclonal antibody. And the other end they are the same. So that end is what we're using for purifying these monoclonal antibodies. So we have these porous beads with a ligand that sits on it that fits very nicely into the standard version of the monoclonal antibody. So that's why these tools are used to a very, very high degree to purify these monoclonal antibodies. And it has a very high selectivity. So for only one step of purification, you get a purity of over 95, maybe 98% purity in only one step. And it also gives you a very quick development time and a quick manufacturing time. What we've done lately is this technology has been around for a number of years. And we see that we need to continuously develop this new technology. It was originally developed together with both the academic and biotech groups in the region. So it's a true example of a collaboration between the academic setting and how it transforms into a commercial setting. We are a very global company, so these technologies are now out there in more than 200 countries and very widely used. It's interesting to see a small academic invention, how it can transform to global use. And we've added on new features to this technology to make sure it's easy to clean so that you can improve the safety of the drugs. And it also, we recently added a new product that has much higher capacity. Because now today, all these cultivation of the cells, they are much more successful. They can grow much more cells with higher levels of producing the monoclonal antibodies. And we've had to improve the capacity of our chromatography resins to cope with that new development. So these are inventions that we, together with these biotech and academic groups in the region, added on to provide new technologies for the industry. So this particular graph shows that with the new technology, with the improved cleaning procedure, you can have these resins for much longer time and with higher safety for producing that particular drug. So this is a true example of how a small technology can have a huge impact to the biopharma industry. So I said it's roughly 90–95% of the biopharma-based drugs use one or more of our technologies. And very many of them use this technology here. So moving to another concept of technologies. So if you have this huge manufacturing facility, if you've seen there's lots of stainless steel tanks, we call them bioreactors, lots of piping. It's very complex to build these facilities. And what we now provide is doing the same thing, but in plastic bags, essentially. So you have a plastic bag that could be up to 1,000 or 2,000 liters. And you can do the reaction in these bags. And instead of cleaning the stainless steel reactor, you just take the plastic bag and throw it away. Put in another bag, and you can start to use the same equipment again, maybe an hour between the batches, and that's it. So we've added speed in the manufacturing. You can also now use the manufacturing facility for a multi-product use, because you can have one product produced one day, and maybe next week you have an entirely new product. In particular, that's helpful if you are a vaccine developer and you want to develop a new strain. And there's new strains for seasonal flu every year. And we also have this sort of pandemics with new threats coming up, which needs to have vaccine treatments. We also see that these technologies reduce the capital expenditure you need to put into this when you build a manufacturing facility. Because you can have the production at smaller scale, you can use the facility much more efficiently, and you can have it for multi-product use. So you can reduce the capital expenditure by half, which will mean that we can build more capacity and serve the unmet medical need. So just one slide to show you a comparison we made about sort of building a traditional manufacturing facility. It usually takes 3, 4, 5 years or so. And if you are, for example, a company in India or China, do you really want to spend 3, 4, 5 years of capital expenditure before you can actually get some revenues in return? No, you'd rather go for something that is disposable, that shortens down that time with about half, and also reduces the capital expenditure by about half. So we see a very high adoption of these technologies in the when you want to build new facilities, because it gives you much higher flexibility, reduces the cost, and increases the speed.